All right, so hello everyone. Today my name is Savannah Draud. I'm a PhD candidate at the University of Mississippi, and I am here today with Dr. Martine Ryberg from Uppsala University in Sweden. Today we're going to be discussing Dr. Ryberg's recent publication called On the Evolution of Ectomycorrhizal Fungi. So just thank you so much for being here.
Thank you. Thank you for having me.
Of course. So my first question that I have for you is, could you talk a little bit about the value of obtaining an in-depth understanding of the evolutionary trajectory of a single partner, in this case the fungal partner, in a mutualistic relationship? And then how well does the information obtained from your research sort of correspond or relate to what we understand about the evolution of the plant lineages that are involved in these partnerships?
Yes. So I guess it's a tricky thing when you investigate an organism that's like so tightly related to another. But when you look at ectomycorrhizae, it's like each relationship is not that specific. So it's not like we see any patterns of like coevolution between a lineage of plant and a lineage of fungi. So it seems like it is a relationship, but it seems like they are evolving like on a large scale, at least independently. But of course, on a smaller scale, you may see unspecific lineages, and so you may see correlations. So therefore, as I see it, like the evolution of ectomycorrhizae in plants is more setting like a framework around, but it's not really restricting what's happening in like when ectomycorrhizae evolve and so on.
Okay.
But in that framework, we check that our results are congruent with like what we know about the evolution of ectomycorrhizae in plants. But as far as I know, it's not as much done in plants actually on this with ectomycorrhizae.
Yeah.
But yeah, from what we know, what we find is congruent. I mean, but some results that we can't react in other ways, we can react by that. It doesn't really make sense in the compared to the evolution of ectomycorrhizae in plants.
Okay, thank you. So the second question is, what do you think accounts for the large diversity of ECM fungal species in comparison to such a small diversity of plant species that participate? And maybe you sort of said this, that they didn't really co-evolve necessarily.
Yeah, so I think that's just a little bit of the big question actually. Like, so what? For me, it's like one of the questions that's amazed me, like the diversity of ectomycorrhizal fungi, and then especially then compared to the diversity of ectomycorrhizal plants. Well, actually looking at the global diversity of ectomycorrhizal plants, it is actually quite a big diversity. But if you look at like single stands, it's usually like few ectomycorrhizal plants and a lot of ectomycorrhizal fungi. And I think, well, yeah, I don't think we really have a good answer on why that is. But so I guess either it's like a niche thing. So maybe you have a lot of more soil niches than you have niches like above ground. Like that could be a possibility, or it could be like maybe you have a lot more individuals of ectomycorrhiza species, and therefore you could have an ectomycorrhizal fungi, and therefore you could have a lot of more species. While for plants, it's not. But I would be, that may not, the difference may not account for the difference we see number of species at least. But it could also be just in a way how they evolve and how new species arise and like which processes drive the speciation and so on.
Right. Okay.
Or a combination of all three.
Lots of options.
Yeah.
And then in your paper, you talk a little bit about like analytical shortcomings associated with studying the evolutionary history of ECM fungi. So could you talk a little bit more about what those shortcomings are?
Yes. So, well, I think we can, when we're looking at this from patterns we see today, and then we're trying to infer what has happened. And the patterns that we see can, there are a lot of different ways that you can interpret these patterns. So the question then is like, how can we decide on which way do we actually believe in that it has happened? And when we do that, we usually when we infer what has happened, we usually use models or use just parsimony. And parsimony, well, I guess it has many known shortcomings, and one of those is that it doesn't account for branch length. Then, so you could if you have a long branch, it's just as likely, or parsimony does not say that it's more likely to have happened changes along that long branch than on a short branch. But also when we use models, we have a lot of assumptions in these models, and I think we need to, if you don't account for are these assumptions reasonable or not, and that's one of the things that I have been looking on them, is comparing these models and comparing different models, and also looking at models accounting for specific changes, so for example, different rates in different parts of the tree and so on. And what we actually found then in another study we did that we refer to in this paper is that, if you don't account for that there's been changes in rates in the tree, well, then we actually get results that are not really making sense, especially then in looking to the evolution of ectomycorrhizal plants, for example. So.
Great. Yeah, and so I really appreciated that in your paper you took the time to sort of explain the different ways that evolutionary data could be analyzed and modeled, and keeping in mind those different shortcomings for each type of modeling. So could you talk a little bit about why you decided to include those explanations in the paper? Because I feel like that's not always something people include.
Yeah, so I guess it may not fit super well in the paper maybe, but it's something that I've been thinking a lot about, and which I think is important, and not only when you look at ancestor strip date reconstruction of one trait in the tree, but also the same type of models are also used now in comparative genomics and so on, and there is a lot of assumptions that goes into those, and the results will totally depend on those assumptions. And so if we don't consider those, we may end up with just the wrong results. Yeah. So I think a deep understanding of the methods is important to be able to interpret the results.
Yeah, definitely. I know it helped me with understanding interpreting what you were telling me later on in the paper. So I appreciated that section. Great.
I'm glad.
So my next question is, so you mentioned that reversals from being ectomycorrhizal back to a saprotrophic lifestyle, or the ability to exist sort of as being either symbiotic or saprotrophic depending on environmental context, those instances are quite rare. But do you think there's something about, something worth studying these rare or unique cases in more depth? And basically, in other words, do you think we should interpret these rarities or exceptions in terms of explaining something about the evolutionary history or trajectory of ectomycorrhizal fungi?
I think it could offer some insights, but it could also be that they are unique things that work in unique settings or unique cases. So I think it's kind of open, if it will tell us something about the evolution in general, or if these are just special cases. But I think understanding how they work at least could make us understand the conflicts between being saprotrophic and ectomycorrhizal, and maybe help us understand the evolution better. But how it will do it, I think, is not given beforehand.
Okay, great. Let's see. So you mentioned that most, but not all, ectomycorrhizal fungal lineages have lost their saprotrophic decay apparatus. Could you talk a bit more about the lineages that haven't lost these traits, and do you think they might represent either a transitional lineage of some sort, or do you think it's more just they're maintaining sort of a unique traits for nutritional flexibility?
I think, there's many people that are much better at discussing these functional aspects, I think. But some of these lineages that have a lot of saprotrophic capabilities, and there are actually some that has been seem to be gaining these saprotrophics within lineages that are that are old lineages of ectomycorrhizal fungi. So it's not just at least that they are transitional. I mean, and I think actually most of those are perhaps not transitional stages. It's probably like actually like some sort of adaptation for specific, having specific functions and be able to do specific things.
Okay. Great. And then could you speak more about the evolution of the effector-like small secreted proteins, or those SSPs, and maybe give me some examples of the orthologs of those genes in the non-symbiotic relatives?
So again, there's other people that are much better at these aspects than I am. But I mean, what has been seen is that many of the things that are expressed when the fungi form the ectomycorrhiza are actually things that you do have homologs like in saprotrophic ancestors or saprotrophic groups also. So you do seem to have other functions in those. Exactly what those functions are is often not very clear then. So for these small secreted proteins, we don't really know exactly what they do in the ectomycorrhiza. We often think that they have some signaling with the plant, and I don't really think we really know what they do in the saprotrophs neither. And it's actually so that even if these specific small secreted proteins that are involved in communication with the plant or like in the formation of ectomycorrhiza, they are usually increasing in the ectomycorrhizal lineages. But if you look at small secreted proteins in total as a family, they are not always increasing. They can even be decreasing in the ectomycorrhizal lineages. So they do have other functions also, and they may have a large set of different functions, is my guess.
Okay, great. Okay, so just moving maybe a little off of the paper specifically, I was hoping maybe you could tell me a bit more about your academic journey and sort of what got you interested in researching fungal evolution to begin with.
Yeah, so well, I've been interested in biology for a long time, and I actually started more of being interested in conservation biology. But I think for many of us, it's like you get a good teacher that's good at teaching at some part. And I had a great teacher in mycology, Niels Hallenberg, who got me interested in fungi. And then I started working on fungi and got into research. And I actually started working on community ecology questions. But I wanted to pursue a PhD, and I got a position working on, I was gonna combine ecological and evolutionary aspects, and that's when I got more into the evolutionary part of it, and it's kind of continued from there.
That's awesome. I think it's great, to change, what you work on as you continue on through your career, because the more background you can get in a lot of different topics, I just feel like it helps you with each new topic you go to. Because I came into grad school originally studying marine biology, and now I'm studying fungi, but I still find connections between the two. So
yeah, and I think it's good to have a different perspective that you bring with you. I think you will be able to see things in a different way.
Absolutely. And then, so what do you think is maybe the most challenging part of the research that you do now, and maybe what do you find most rewarding about it?
Well, starting with most rewarding, I mean, for me, it's the curiosity and getting new insights and learning. I think it's just in a way it's a continuation of being a student. You learn all the time, and learning new things. That's kind of the drive for me. When you sit there and you realize, oh, this is how it works, like the eureka moments, I think are the drive. The challenges, well, I mean, the challenges are also part of the fun, I think, in a way, trying to solving things. I think a lot of the challenges are perhaps in the things surrounding the science. So administrative challenges, and when you get to be more senior, you get the supervision and how to supervise students the best way. And I think those are probably the most challenging parts.
Mhm.
But of course, you have new techniques coming and understanding like all of these different proteins that you're getting into now, like MISPs and Casymes and all, learning new science. That's of course challenging, but it's also interesting, I think, and one of the things that are most rewarding.
Yeah. I like that you said the challenges can be some of the most rewarding because I feel the same way. Like I get very frustrated by something that I'm trying to learn, but then when I finally figure it out, it's like the best feeling in the world. So I
Yeah,
Okay, and then my last question for you is going back a little bit to the research itself. Like, what do you see for the future of this kind of research? Like the next big questions, or maybe questions that need to be readdressed?
I think we're still, or for me at least, it's still this question about why do we have so many ectomycorrhizal fungi, and then especially then in relationship to the plants in the community. I think that is still the big question, and I don't think I will be able to answer it during my career, perhaps, but I hopefully will be able to get a little bit closer. So for me, that is actually the big question. And I think, well, now I guess is compared to genomics, it's kind of well established, but I think now we have like these big data sets, and it's trying to make sense out of all of this data and bringing it together. I think that's the biggest challenge, perhaps, to handle all of this data.
Great. Well, is there anything else that you'd like to add, or something that I should have asked you?
No, they were great questions. So I do not think now. I don't think I have anything additional.
Okay. Well, thank you so much. I really appreciate you taking the time to speak with me and share your research with us.
Great, thank you, and thank you for having me again. Yeah.
Yeah, of course.